Okay, so today we're going to do the review for Lab Test 1, and Lab Test 1 is going to be Thursday at 8:35, which is your regular lab test time. And what you need to bring that day is a really good pencil with a good eraser, because we don't have pencil, we don't have pencil sharpeners in the building, so make sure you're prepared for that. And then you're going to need a calculator, because you can't use your cell phone. When you come that day, the lab will be locked, so you just have to hang out in the hallway. And then when I let you in, make sure your cell phone is turned off and everything's put in your backpack. I wouldn't recommend you bring a beverage that day or complete your beverage before you come into the room, because this is what we call a lab practical, which means we move around the room. Many of you have taken lab practicals before, so you understand that trying to carry a beverage, a calculator, a pencil, and a Scantron all at the same time gets a little challenging, okay? So I wouldn't recommend it that day. Okay, so we're going to start with experiment 1, the scientific method. And again, if you don't have a review sheet, I have extras here in the front. So I'm just going along with your review sheet, and it says outline the steps of the scientific method. And so let's go ahead and outline the steps of the scientific method. What's step number 1? Observation, that's good. And if you're wondering where am I getting this from, I'm getting this from a handout that I gave you from the day that we worked on earthworms. But if you don't have that, this is also in your lab manual, page 3. So observations, which could be visual observations, they could be what you hear, what you feel, etc. Okay, step number 2. Hypothesis. Okay, what is a hypothesis? An educated guess, also a tentative explanation. And the hypothesis is based on your observations. Don't forget, if you don't have a review sheet, I have them up in the front. Okay, from your hypothesis, what is step number 4? Say it again, Alaska. Or Emily. I can't hear. Experiment, also known as testing. So if you thought of testing, that's correct. And then what's the last step of the scientific method? I'm sorry? Last step of the scientific method? Thank you very much. Conclusion. Now don't forget that your conclusion can either support or not support your hypothesis. That's all conclusions do, is they support or not support the hypothesis. Okay, so those are the 4 steps of the scientific method. Anybody have a question about number 1? Super. Okay, number 2, I just got this from your review sheet. If given an experimental scenario, be able to determine the experimental group, control group, experimental variable, and data. So, experimental group. Okay, so you recall we did an activity, or we talked about an activity where we were trying to figure out if the algae that shrimp ate were affecting their color. And you recall, we did this in lecture, that the groups that we were feeding, the different kinds of algae, the red algae, the green algae, the brown algae, those were our experimental groups. But we had a 4th group that we fed shrimp chow. Okay, what group would you call that? The control group. And we have always have controls in our experiments. Remember, in a control group, it's exactly set up like the experimental groups, but you should already know what the outcome is going to be on a control group. Okay, now controls are there for 2 purposes, and if you missed this on the lecture test last week, then don't miss it again. Controls are for 2 reasons. Control groups are for 2 reasons. What are the 2 reasons you always have a control in an experiment? Say it again, Emily. There's a specific word. It starts with the letter V. Sorry? Validity. It validates your experiment. So if your control doesn't come out as you expected, that means your experiment is not valid. Okay, and then what's the second reason we have controls? This is the more practical reason. Comparison. Excellent job. So everybody has to know the two reasons that you have a control and experiment is validity and comparison. That's why you have to have a control in experimental procedure. Okay, in our shrimp experiment, remember we had 3 tanks that we fed different kinds of algae. The 4th tank we fed shrimp chow. What was our experimental variable? That's what was different tank to tank. So what was our experimental variable? Their diet. Excellent choice of words. Their diet. That's very good. And then you may recall that we collected information, or we would collect information on how many died, what color they were at the end of the experiment, what the pH of the water would have been, what the dissolved oxygen in the water would have been, what the temperature of the water would have been. What do you call all that information that you collect during an experiment? Data. Okay, so everybody take a look at the 4 dots I have up here. Is everybody okay on how you can tell an experimental group from the control group? And you know the 2 purposes of the control group. You could identify experimental variable, and you could tell me what data is. It's okay on what's up on the screen right now? Okay, super. Okay, be able to form a hypothesis. And so we practiced that a little bit the day we were working with earthworms. I remember we wrote some hypotheses, so I would like for you to go back and review that activity that we did on earthworms and notice how we wrote a hypothesis. And at the end of the activity, I think I gave you statements, and you had to pick which of the statements was a hypothesis. So go back and practice that, please. And then reaching a conclusion. I have no fear that you're going to be able to do that, because every time we've been doing an experiment, we have been reaching conclusions. You guys may recall every time we do an experiment, at the end we stop as a class and talk about it, and then we reach a conclusion based on the data that we have collected. Okay, so did anybody have a question about the scientific method? Okay, great. We're going to move on to metric measurement. And metric measurement, we actually started metric measurement whenever we were our first day together. And metric measurement in your laboratory manual is on page 10. In case you want a reference page for metric measurement, page 10. So I have know the meaning of the metric prefixes. And so guys, I put some stuff up here on the screen to see if you could fill these blanks out. Okay, take a look at what I have up on the screen, and I want you to write these down or put them into your notes. You should be able to fill in the blanks. As you're filling in the blanks, if there's anybody who needs a review sheet, I have extras here in the front. Okay, so let's go ahead and get started. Does anybody have how many centimeters are in 1 meter? Say it again. 100. Who told me that? Excellent job. Isabel. Good, Isabel. So it's 100. Everybody okay on that? 100 centimeters equals 1 meter. Everybody okay on that one? Yes? Okay. How many millimeters in 1 centimeter? 10. Who told me that? Tim. Good job. So then it's simply doing math now. How many millimeters are in a meter? Let's do it again. 1,000. Excellent job. Sorry, I have old ears. I can't hear. Okay, now, one day we were putting a ruler under the microscope and looking at it under the microscope, and here's what I remember. We could see some millimeters when we were on 4x. Then we saw 5 of them. And then we went to 10x, and we could see 2 of them. And then we went to high power, and we couldn't see a millimeter at all. Remember, we just had to guess. We thought it was about 1/2 of a millimeter. And then I think Caleb figured out, if I remember properly, that he figured out we could figure it out mathematically. Wasn't that you who figured it out? And mathematically, we did figure out that the field of view on high power was only 1/2 of a millimeter. So everybody take a look at this next screen and write this down. So it didn't take very long for you and me to figure out that millimeters are simply too big to use as a unit of measurement when you're using a microscope. Because we figured it out. So I'm figuring out that microscopists figured it out a long time ago, that millimeters are simply too big to be very useful when you're measuring stuff under the microscope. So they introduced a new term, and it's called a micrometer or micrometer. Here, I'll spell it for you at the bottom. I pronounce it micrometer. If you'd like micrometer better, that's cool. Does everybody see why they couldn't use MM as its abbreviation? How come they couldn't use MM? It's already taken. So this fancy U right there is the abbreviation for micro. It's an abbreviation for micro. So this is micrometer. Okay, so they figured out that millimeters were too big, so they divided 1 millimeter into 1,000 equal units, and they decided to call those units micrometers. And we use micrometers to measure things under the compound light microscope. That's kind of our standard unit. And that was working out real good until we developed the electron microscope. And you guys remember the electron microscope magnifies things even more. So the micrometer got to be too big to be useful under the electron microscope. So they developed this next unit. Everybody see my next one, NM? Does anybody know what the N stands for in NM? Nano. That's very good. N-A-N-O. And they divided a micrometer into 1,000 equal units, and we call those nanometers or nanometers. And again, N-A-N-O. In case you don't know what I'm saying, it's N-A-N-O. So NM is nanometers or nanometers. Okay, so see if you can do the math here. I want to know how many, I already have that, this is wrong. I want to know how many nanometers there are in a millimeter. That's what I want to know. How many nanometers are in a millimeter? What'd you come up with? It is. Good job, Tim. How'd you do it, Tim? He put all the zeros together, and then Alex is right. He is a genius, yes. Very good, Alex. Glad you recognize it. So he did 1,000 times 1,000, which means he had to put a 1 and then all those zeros. If you count all those zeros, that's 6 zeros. So my point is nanometers are pretty dang small. You thought I was going to cuss. They're pretty dang small. Yes? It's okay with nanometers. So let me tell you when these would be useful. Micrometer are really good for measuring cells. And we've been looking at cells under the microscope like onion cells, the nucleus of an onion cell, your cheek cells. It's really useful that way. Whereas nanometers are useful for measuring things with the electron microscope when the magnification is really, really high. So it really just depends on what instrument you're using, on what unit you would use. Is everybody okay on using these units? Super. So let's see if we can do some conversions here. This goes with question number 2. Okay, now remember from the previous screen that you told me that 10 millimeters are needed to make 1 centimeter. So how many millimeters are there in 23 centimeters? 230. Good job, Alex. We okay on how Alex got number 1, or the first one? Everybody's okay on that one? Okay. Now I caught, oh, not now. On this one, you shouldn't get a decimal. So let's talk about the, are you talking about the second one, Jordan?
Yeah.
Okay. So let's put first what we know from the previous screen. We said it takes 1,000 micrometers to equal 1 millimeter. Does everybody see I got that from the previous screen? Okay, so Jordan's right. We do have to move the decimal. And does everybody agree we have to move it 3 times? But do we move it to the right or do we move it to the left is the question. Okay, so would you just answer my question though? Do we need to move it to the right or move it to the left? This is logic. We don't have to look at a book. This is logic. To the right. We have to move it to the right. Because look, to get 1 to turn into 1,000, you're going to have to move the decimal to the right. Move it to the right. So I'm going to have trouble fitting it into this little space, but what is the answer?
368,000.
Does everybody agree with Lindsay? Everybody see how this is logical? You don't even need a book. This is logic. Okay? Okay, so let's try this last one. And again, I'm going to write down what we knew from our previous page. We said that 1 micrometer or micrometer was 1,000 nanometers or nanometers. So what would 6 micrometers be? I'm sorry? 6,000. Everybody see how logical this is? Okay. They have a question about what we have on screen right now. You're okay on the conversions? Okay, great. All right, I want you to open your drawer. Buddy, open a drawer and find yourself a little plastic ruler. You recognize that plastic ruler. We use it most every day. Look for that little plastic ruler that we use most every day. Is there anybody who doesn't have that little plastic ruler? Come on up. Anybody else need a plastic ruler? Okay, so we're looking at the metric side. Make sure you're not looking at the inches. You're looking at the metric side. And so I have look at the ruler, and you should be able to figure out how many centimeters are on the ruler and how many millimeters are on the ruler. So how many centimeters are on this ruler? What? Was that my genius? Ah. Everybody agreeing with Tim? 15? Yes? Okay, we're going with 15. Okay, now how many millimeters are on this ruler? 150. Who told me that? Very good. Stephania said it is 150. How'd you do that, Stephania? Multiply it times 10. Or for those of you that are big decimal movers, you just move your decimal 1 time to the right. But okay on looking at the ruler. Super. Hey. What are the freezing and boiling points of water? Now this has to be in Celsius because this is a science class, so this has to be in Celsius. Okay, go ahead and give me the freezing point of water in Celsius. 0 degrees. That's good. That's Katie Messina. 0 degrees Celsius. And boiling point? 100. Good job, class. Okay, on the freezing and boiling points of water. If you need a review sheet, I got extras up here in the front. Okay, the day that we first met, our very first day together, you had to do some measuring. And so I'm going to tell you again what you're going to have to be able to measure on a test. Using a ruler, you're going to have to be able to measure the length of bones. Everybody okay on that? Make sure that you're using the metric side of the ruler. And then you're also going to have to weigh a bone or something. You have to weigh something on the triple beam balance to get its mass in grams. Everybody okay on that idea? Being able to use the ruler and the triple beam balance. Okay, and then we did volume 2 different ways. There was a seashell and there was a cube. And we did their volumes in 2 different ways. Does anybody remember how we did the volume of the seashell? Good, we use water in a graduated cylinder. Now, do you have to put water in the graduated cylinder before you put in the seashell? Yes. And do you have to know how much water is in the graduated cylinder before you put in the seashell? Yes. Okay, it's your choice how much water goes in there, but I would recommend enough water to cover up the seashell. Okay, so you have to put water in the graduated cylinder first. You have to know how much water's in there, and then you drop in the seashell. Then what do? Okay, so say it again louder, Alaska.
You measure the new water level and then subtract it out.
She said you measure the new water level and subtract it off the old water level, and then you get your answer, and then the unit would be milliliters. So I'm just making it up. Let's say it was 28 milliliters to start with, and it went up to 33 milliliters. What's the volume? 5. Your math skills are excellent. Yes, 33 minus 28 is 5 milliliters. So everybody's okay on doing volume using the graduated cylinder? Yes? Okay, and then the cube was a little boggle, piece of wood cube is a little cube. How did you do volume on that one? Length times width times height in centimeters. And that's how we got the volume of the cube. That's why you need a calculator on Thursday. Okay? Length times width times height. But okay, I want to have up on the screen right now doing length, doing weight, and doing volume 2 different ways. Super. Okay, I have what is a meniscus? And I have a little drawing there to help you remember what a meniscus. I know it's a good one. What is a meniscus? That's good. Stephanie said it's the way the water is concave in a graduated cylinder. You guys know why. Because water's adhesive to the glass, remember? So it's sticking to the glass. So how do you properly read a meniscus? You read the bottom, and you need to get eye level. Now, guys, here's what I don't want to see on the test. I don't want to see you picking that graduated cylinder up to read it. I don't want to see that. What do?
Get eye level.
Get eye level. You got to leave it on the table, and then you get eye level to read it. Now, let me tell you, you got to really know what you're doing because it's a multiple choice test. And so, again, I'm just making this up. Let's say there's 32 milliliters of water in that graduated cylinder. I'm going to have 30 as a choice. I'm going to have 31 as a choice. I'm going to have 32 as a choice. I'm going to have 33 as a choice. So you're going to have to know what you're doing. Okay, so I'm going to have all the ballpark choices there. You really have to know what you're doing. Everybody okay with on the meniscus? Yes. Oh, good question, Stephanie. She said, how many questions are on the test? There are 50 multiple choice questions on the test. So that means that they're worth each about 1.8 points each, because your quiz and your unknown that you did a few weeks ago, you remember taking a little quiz and you did an unknown in a little vial? That's worth 10 points. Those two are worth 10 points. So that the test that you're taking Thursday is worth 90 points, and then the other activities that we have done so far are worth 10 points. So they all combine for one test grade. It's an excellent question, Stephanie. Okay, so we're going to leave the metric system unless anybody has a question. If you see anything you need repeated, just raise your hand. I'll repeat for you. But we're going to go over to microscopy. And this is on page 14, if you're keeping up with where we are in our lab manual. Sometimes reference pages will help you when you go back to review. So exercise 2, we started on page 14 in our laboratory manual. Okay, I have what kind of microscope would be used to view? Okay, so let's take a look at that. Page 14, if you just wanted to look at, let's just say, I don't know, your cheek cells, then you would just use a compound light microscope. But what if you wanted to see the ribosomes in your cheek cells? Does everybody agree that your cheek cells have ribosomes? How do you know your cheek cells have ribosomes? Because all cells do. Good job. So if you were at a place that had anything that you needed to study cells, not here, but someplace else, like, I don't know, UT, A&M, University of Houston, and you wanted to look at the ribosomes of your cheek cells, what would you use? An electron microscope. That's good. Okay, so if you just wanted to see your cheek cells, a compound light microscope would be sufficient. But you can't see ribosomes under the compound light microscope. How come? They're too small. Sure, they're too small. So then we'd have to have something with higher magnification, an electron microscope. That's good. Okay, let's just talk about sand and seashells. We went to the beach and we collected a bunch of sand, seashells, and some seaweed, and we wanted to look at the sand, the seashells, and the seaweed in more detail, just to see it better, see what the surfaces look like, you know, what do these things look like in more detail. What kind of scope would we use? It would be a light microscope. Would it be a compound light microscope? No. There's another one. Very good. It's the stereo microscope. You guys remember using it before. It's kind of like a really fancy magnifying glass. We looked at our earthworms using it, and then one day we walked around and looked at all kinds of things. Some of us looked at feathers, others looked at moths, some of us looked at plant material. So everybody remember the stereoscope? Stereoscope. That's okay? All right. So I have, how are light microscopes and electron microscopes different? Okay, so light microscopes have lower magnification than electron microscopes. Is everybody okay on that one? Yes? All right, and then just real quickly I want to tell you that compound light microscopes, like that are in that cabinet over there, you've used them several times. Everybody with me on what a compound light microscope is? That's when you want to look at the internal details of something, like you want to see inside your cheek cells. Whereas, as Lindsay mentioned, the stereo microscope, that's where you want to look for surface details. Surface details. So some scopes show you internal details, and some scopes show you surface details. Compound light microscopes are for internal details. Stereoscopes are for surface details.
So the light microscope is for what?
Internal details. Very good. Okay, just as a side note, electron microscopes come in two flavors also. They'll show you internal details, and then they'll show you surface details. I'm just going to mention them. They're not on your test. I'm just going to mention them, but these are not on your test. Electron microscopes that show internal details are called transmission electron microscopes, and you'll see many pictures in your textbook from transmission electron microscopes. Like, if you will, page 14, letter B. That's from a transmission electron microscope. See how I can see inside of that cell? Whereas the scanning electron microscope shows you surface details like letter C on page 14. So light microscopes, you get two choices: internal details, compound light microscope; surface details, stereoscope. And the same thing happens with electron microscopes. Between compound light microscope and a transmission electron microscope, which one could give you higher magnification? A light microscope or an electron microscope? Oh, for sure, electron microscope. So electron microscopes can be used to really see small things, really small things. It really opened up a whole new world when those were developed. We could really see small things. Just want to comment, like today. Everybody knows today, if you listen to the news today, you're going to hear about a virus in Africa. What's the name of that virus? Ebola. And how do you think they're visualizing it? How do you think they know what it looks like and what it's made out of? The electron microscope. Yeah, you can see viruses using an electron microscope because viruses are so small. Okay, that's good. Okay, know how to use a dissecting microscope. And a dissecting microscope is also called a stereoscope. So I would just ask you to be able to change its magnification or to focus it or change the light. What can you view with it? Anything you want to. If you want to see the surface details, you can use it to look at anything. We used it to look at feathers, to look at plants, to look at insects, to look at worms. You can look at anything you want to. You're only going to see surface details with the stereoscope. That's all you're going to see is surface details. So again, stereoscopes are like fancy magnifying glasses. I have to tell you, when we first got those scopes, I was teaching genetics, and we were doing fruit flies. My genetics students were crossing fruit flies. You know, we'd know this was the dad and this is the mom, and we let them mate, and then we take the parents out and collect all the offspring and look at them under the stereoscope and count how many came out looking this way or how many came out looking that way. And the details on those flies underneath the stereoscope are amazing. You can see every facet of their eye, every hair on their body. It's pretty amazing. Okay, number 4 should be starred. There will be many questions about number 4 on your lab test. The compound light microscope is on page 18 and 19 in your laboratory manual. I'm going to put some stickers on the parts of the scope. I'm just making this up. I'll get a sticker number 18 and stick it on the ocular. And then you have to say, well, this is called the ocular, and you have to find ocular and color that in on your Scantron. Or maybe I'll put the sticker on the ocular, and the question will say, what's the function of the ocular? And then you'll have to put to magnify and view objects. Because you have to know what's the name of the part and what's the part used for. Understand that question, what's being asked of you. Okay, so several questions on that one. Great. Okay, know how to use the compound light microscope on low and high powers. So everybody know you always start on 4x? Always. Why do we always start on 4x? Because it's got the biggest, what's that called? Yeah, it's got the biggest field of view. You know, that circle of white light is called the field of view. And it's got the biggest field of view, so you can see the most slide on 4x. 4x. Okay, so you're going to focus and center on 4x, then you're going to go to 10x, focus and center on 10x. Sometimes that's as far as we go. And then if we go into high power, then we move on to high power. Now, when you get to high power, there's one adjustment you cannot use. What can you not use on high power? The coarse. Good. You cannot use coarse adjustment. You can't focus with the coarse adjustment on high power. What can you use to focus on high power? The fine adjustment. That's the only one. Okay, so here's my idea. I need to know as a student that I always start on 4, focus and center, go to 10, focus and center, go to 40, and when I'm on 40, all I can use is fine adjustment. Everybody's okay on that idea? Super. Be able to calculate total magnification. Well, we did that together on page 21. I want to reference page 21, table 2.3. What's the basic calculation for total magnification? What do you do? Huh? I didn't hear you. I'm still waiting. Guys, the test's in 2 days. What are you going to do?
The ocular lens is 10x, so whatever power you have it on, you times it by that. That's your magnification.
Yeah? Ocular times objective. Everybody agree? You don't have to even memorize what they are. How do you find the numbers on the microscope? Look for the number that has what after it? The X. And then the X even reminds you to multiply. How sweet is that? So you're going to look and see what the ocular is, and then look to see what objective is clicked into position. And you just multiply those two things together. Ready? Okay, on total magnification. It's yes? It's great. Oh, we already know the answer to this one. Which objective would give the largest field of view? Scanning, 4x, also known as scanning. And we even proved that, I remember, with our ruler one day. Okay. All right, what is inversion? Inversion, everybody's on page 20 in the middle of the page. In case you need a reference page for inversion. And here's what I remember we used to show inversion. What is that? That's the letter E. And this is what the letter E looked like for most of us in the classroom before we mounted it on the scope. What did it look like after you mounted it and viewed it under the scope? Looked like a normal E, and I remember also it was bigger. Okay, so everybody check it out. This was the before, and the before, that was the top of the letter E, and now where is it? Now it's down here. And this was the bottom of the letter E, and now where is it? The top. This was the left part of the letter E, and now the left part is on the right. And this was the right part of the letter E, and now the right part is on the left. So everything is totally flipped. Bottom became the top, left became the right. Everybody okay with that? So this was before, and this was after. So everybody okay with the idea of inversion? Okay, super. How does inversion relate to microscope usage? So it's kind of like, well, who cares? So why should you care that the microscope inverts images? Like, what's the application of that? Why do you care? It's going to change the way you see things. It's good. What else?
When you make your notes or whatever, you have to know where it actually is, so knowing that it's inverted would help you to figure out how it actually is.
So how it actually is. That's good. Okay, how about when you start moving your slide? If I move my slide to the left, does the image go to the left? No. Which way does it go? To the right. So that all movement under the microscope is opposite. So I'll write that down for you. Movement under the scope is opposite, and that's because of the inversion. Up goes down, left goes right. Is okay on that? Super. Okay, does anybody have a question about exercise 2 on microscopy? Nobody? Okay, we're going to go to exercise 3, and most of what we did on exercise 3 is summarized for us on page 40. Lots of what we need is on page 40. So would you flip over to page 40 in your laboratory manual? So exercise 3 is the chemical composition of cells. Okay, class, what chemical is used to test for protein? Thank you, Karen. Biuret. I need the positive color in the test tube. What color are you looking for that means yes, there's protein? Everybody see purple? Yes? Or kind of a pinkish purple? Everybody see I'm getting this off page 40? Yes? Okay, what's the negative color? Blue. But okay on protein. Okay, I have what is used to test for starch? Good job. Iodine. Positive color? All the dark colors. I'm talking black, navy blue, dark purple, chocolate brown. Like dark chocolate brown. Okay, what's the negative color? Good job. Amber. We have a question about starch. Great. What is used to test for sugar? Benedict's. And what else? Yes, I'm going to put heat here. You're right, we had to heat it. Positive color? That's right. Green meant a little bit. Yellow meant quite a bit. And orange is, oh my gosh, there's so much sugar in there. What's the negative color? Blue. Good job. So yes, we memorized this for the quiz, but we still have to know it. Okay, we still have to know it. Okay. How do you use brown paper to test for lipid? Put it in the microwave. Okay, we're looking for, is the word transparent? Thank you. We're looking for light to be able to come through. We're going to call that translucent. We're looking for a translucent spot. If we see a translucent spot, then yes, we have lipid. And if there's no translucent spot, then we are negative no for lipid. And I need you to be able to do that on the test also. And feel free to hold things up to the light. And another thing, side comment on these previous screens, on the sugar, on the iodine, the Biuret, I'll have a negative control for each of those. I'll have the test tube that I'm asking you about, and then I'll have a negative control next to it so you can compare. Because all experiments have controls, okay? But you feeling confident on that one? Super. All right, so now I want to just reflect back to a few other pages that were in this lab. I'm going to give you some page number references. Number 2 I found on page 28. Bless you, Kay. Page 28. I have, what are the building blocks of carbohydrates? We're on page 28. Anybody? Building blocks of carbohydrates. Glucose is an example. Simple sugars. Now, guys, simple sugars is good. I'll take that. Thank you. Who told me that? Monosaccharides. Is it Isabel? Good job. Monosaccharides. So, like here's how it goes. First thing that pops in my head is simple sugars. Now I look at the choices and it's not there. So there you go. You go, darn. So then you look at the choices again and you find monosaccharides. They are the same thing. They equal each other.
Is that a hint that it's not going to be simple sugar on the test?
Say it again, Tim.
Is that a hint that it's not going to be simple sugars on the test?
That's a very strong one. Thank you very much. Yes. See, he is. I'm telling you. He does well. Okay, building blocks of lipids. Let me help you guys out with the page number. I found the building blocks of lipids several pages over. Page 34. Page 34. Building blocks of lipids. Page 34.
Glycerol and 3 fatty acids.
Thank you. Glycerol plus 3 fatty acids. Glycerol plus 3 fatty acids. And that's page 34 if you need a reference page for that question. So lipids, page 34. Okay, building blocks of proteins. Man, I don't have these in order very well for you. Sorry. Page 33, back a page. Page 33 for the building blocks of proteins. But you don't even need a page. Everybody knows this one. Well, sure enough, amino acids. Is okay? Yes? Excellent. Okay, now let's kick it up a notch. There are so many page numbers we could reference for this question, but I'm going to get you to go back to page 28 for this question. It says, how are these monomers combined to produce polymers? And the example that's on page 28 are the monomers called monosaccharides. And they're being combined to form a disaccharide. And I want to know, what do you call that kind of chemical reaction where 2 small molecules, oops, I don't know what just happened. There it is. 2 small molecules are combined to make a bigger molecule. Dehydration. That's excellent. Good to know. You okay on that question? Now look, guys, you could look on page 28 for that, but check it out. Same thing happens on page 33. Look on page 33. Instead of combining two monosaccharides to make a disaccharide, I combine two amino acids to make a dipeptide. But isn't it the same thing? Yes? Check it out, page 34. When I threw three fatty acids together with glycerol to make a fat, look, I was doing the same thing. Same thing. Okay, great. Okay, how are polymers broken down to produce monomers? What kind of chemical reaction are you doing? Thank you, hydrolysis. And again, any of those page numbers would be great references. That was all question 2. Okay, question 3. Define a monosaccharide. We already did. What's the answer? Thank you. Simple sugar. And we actually already have an example that's been mentioned in class today of a monosaccharide. Glucose. Everybody okay on that one? Great. I need a disaccharide. Maltose is an example. I'll take it. How do you define it? 2 simple sugars. That's good. Or maybe you were thinking 2 monosaccharides. That would be great also. Same thing. Everybody okay on that one? Okay. Define polysaccharide. Thank you. Many. Need an example.
Starch.
Thank you. Who is that? Eric. Good job. Starch. Some of you may have thought of cellulose. That's a good one. You don't have to know all these, but I'm just throwing some out there that you may have studied in your lecture notes. Some of you might have thought of chitin, that really cool stuff that is used to make the exoskeleton of arthropods. Some of you might have thought of glycogen. These are some that we mentioned in our notes before. But most people think of starch. That's a good one. Starch. I have, what is the common, that's a good question. What is the common energy storage form of glucose in plants? Well, first let me tell you guys that this letter, capital letter E, I'm going to start using it today. It's my abbreviation for energy. So anytime you see me use just the capital letter E, it's going to be the energy. We're going to start using that for Chapter 5. Most of you have done the quiz on Chapter 5, so you know energy is a big part of that chapter. And so we'll start using capital E for energy. Anyway, what's the common energy storage form of glucose that you find in plants? Anybody know the answer to that? It is starch. Good. That's how plants store their glucose that they're going to use for energy later down the road. That's how they store it, as starch. And that's why we like plants that have lots of starch, because they're full of energy. Okay. What plant structures store starch? This question's so easy, I can't even believe I asked you this. What's the answer? Look how easy that is, huh? It's so easy you would be afraid to pick it as a choice, wouldn't you? But look at the question. What plant structures store this substance, which is known as starch? Starch grains. Are you okay on that one? Now, you and I saw starch grains under the microscope. Does anybody remember what plant tissue we made a tiny slice of to see starch grains under the microscope? Potato. Which brings us to the next question. What must be added to potato cells to see starch grains? Thank you very much. Iodine. Good job. Great. Number 5's harder. It's a little harder, I think.
What is it?
Yeah, what is it? Question number 5. What is the structural form of glucose? It's a polymer, and plants have it in their cells, but they don't use this for energy. They use it simply for structure. It is cellulose. That's good. This one's cellulose. What do most people call cellulose? Fiber. Very good. It's the common name of cellulose is fiber. But okay so far? Great. What cell part, this would be a plant cell part, what plant cell part is composed of cellulose? Cell wall. Good job. In plants, the cell wall is made out of cellulose. Should you have cellulose in your diet? Yes, you should. Give me 2 reasons. Very good, Jordan. Weight management. It makes you feel full so you don't eat things that are really high in calories. What else? Keeps you regular. Keeps the food moving through your food tube. That's real important. But you okay on this question? You're okay? Okay. What term describes the bond between amino acids in a protein? Good, Emily. Peptide bond. Look, and if you didn't know that, that didn't just immediately come to your mind, let me give you that page number again. Page 33 is a good reference for peptide bond. If you want to go back and look at that, that's page 33, peptide bond. Okay, number 7. I'm going to give you 2 test tubes of lipid. Okay, let me just draw this for you. I'm going to give you 2 test tubes of lipid. And I'm going to put stoppers in them because I am going to allow you to pick them up and shake them. Okay, so they're going to have stoppers in them so you don't make a big mess. I've seen that happen. Shake it. Okay. I'm going to put liquid in both of them. Then I'm going to put in some oil. Now I'm going to allow you to shake them. Okay, and we'll call this test tube 1 and this test tube 2. Let's say that you shake it. And on test tube 1, after you shake it and you put it back, well, you finish shaking it and you look at it, it separates back like that. But test tube 2, when you shake it, instead, you see little bubbles in there. Okay, which one was oil and water? Test tube 1 or test tube 2? Test tube 1. Okay, and which tube was oil and an emulsifier? Test tube 2. So just for a quick reference on that, we did that together, this activity, on page 35 and page 36. Page 35 and page 36 are great pages to reference, to go back and review when we did the 2 test tubes and how we shook them and looked to see what was going on. You're going to have to do that on the test. And again, feel free to pick the test tubes up. Any of the test tubes during the during the test, you're allowed to pick up the test tubes and hold them up to the light. That way you get really good colors, you can see really well, you can see the bubbles, etc. So feel free to pick up the visuals and look at them during the test. Okay? Video count number 7. Okay, so everybody agreed test tube number 2 had the emulsifier. Okay, great. All right, adipose tissues on page 37. Page 37, adipose tissue. So adipose tissue kind of looks like bubbles. There's, a much better picture on page 37, everybody. And then you can kind of see on the sides of the cells, you'll see the nucleus and cytoplasm. And of course, what's taking up the middle of the cell literally is fat. Yeah, these are fat storage cells. Adipose tissue. Okay, I need 3 functions of adipose. Go ahead.
Insulation.
Good. Keeps us warm. Insulation. Energy storage. Because fat is excellent energy storage. Can you remember, this is on the test last week, can you remember how many calories per gram fat has? 9. It's excellent for energy storage. And then I heard the third function. What was it? Protection. It's all cushy. And I think you probably already listed these in your lab manual, page 37. A great reference page number is page 37. Where can you find adipose in your body? Oh, please. Where can you find adipose in your body? Okay, so around organs, and there it's doing protection. Where else?
Muscle.
Say it again. You can find it around muscle. Sometimes if you're super fat, like a cow or a pig, because we fed them so much to make them fat, then you'll actually find the fat in the muscle. Okay, so around muscles, yeah. But mostly, on most animals, where do you go to find fat? Under the skin. Mm-hmm. Some of you have dissected, and so you've seen fat in animals and where it's at. If that's something you've never experienced, make sure you take General Biology 2. So we do dissection there, and the big animal that we dissect is a fetal pig, which those guys haven't really had the time to get too terribly fat, but you'll be able to see where the natural deposition of fat is, even for the leanest animals, where you can find the adipose at. Okay, so that should take care of exercise 3, chemical composition of cells. Did anybody need anything repeated for exercise 3?
Question
Number 7? Okay, so on question number 7, we'll have 2 test tubes, and Karen, you'll be able to pick them up and shake them and hold them up to the light.
What was the page?
Oh, the page number, I'm sorry. Page 35 and 36. Anybody else? All right, super. So exercise 4, we actually worked on exercise 4 for 3 different days. So expect quite a few questions here. Number 1 is know how to determine if a cell is prokaryotic or eukaryotic. And guys, we kind of did this on page 40 at the bottom of the page. Page 40, look at the bottom of the page, where you should have done some sketches and labeling. So using a microscope, how would you be able to tell if an organism is pro or eukaryotic?
Pro had no nucleus.
Good. Pro, no nucleus.
Pro, I think, is autotroph.
That one that we looked at was an autotroph. Now, thank you for that comment, Jordan. There will be nothing on the test that you have not looked at. However, everything we looked at will be on the test. Yeah. But I'm not going to throw new stuff on there that you've never seen before. Okay, so if we made wet mounts out of it and we looked at it under the microscope, then expect it to be on the test. So make sure you consider all the sketches that you did. Make sure you look at all of those. Okay, so prokaryotic means no nucleus, and of course eukaryotic does have a nucleus. And I really think that page 40 is a nice example of that, because we did the oscillatoria, which had no nucleus, and when I was asking you, I remember some of you were a little tentative. You weren't really sure. You wanted to say there wasn't a nucleus, but you really weren't sure until we looked at our cheek cells, and you could see the nucleus in your cheek cells quite clearly. And then I think you were more convinced that the oscillatoria indeed had no nucleus. A nucleus is a big structure, quite obvious in a cell. Okay, so everybody feels comfortable being able to do that? Okay, now Jordan's comment also is relative. You have to be able to tell if something's autotrophic or heterotrophic also. It's a giveaway that it's an autotroph if what? Okay, cell wall. If you see a cell wall, that's good. Anything else? Green, that's good. I have what characteristics do all cells have in common? Give me some things all cells have. No, who said nucleus? Plasma membrane, I'll take that. And that is one of our topics today in lectures, the plasma membrane. What else? Ribosomes to make their proteins with. What else?
DNA.
Thank you, DNA. So the three things we've talked about so far that all cells have. All cells have these three things. Be able to identify the cell parts of an animal cell. And the animal cell that I want to use, of course, are your cheek cells, animal cheek cells. So review that sketch on page 40. And everybody, page 44, you need to go back and review page 44, which I asked you to complete last week prior to our lecture test. Page 44 would be a good page to go back and review your animal cell parts, page 44. And then plant cells on page 45. Page 45. Go back and review page 45. If you haven't done those two pages, then that's too bad. Because those of you that did those pages, remember last week, and I came around and I wrote down who did and who didn't do those? If you did them, you got 4 points on your lecture test last week. And if you didn't, then you didn't get 4 points. So some of you got a 4-point bonus, some of you did not. So there you go. Page 46. Page 46, we did three different wet mounts of plants. I need you to go back and review those, the onion, the potato, and the elodia. Make sure you review that they're plant cells and be able to identify the parts that we labeled. Page 46. Now the elodia are pictures on page 46. And then the onion and the potato were sketches at the bottom of page 46. So all of our plant cells are on page 46. Page 46. Okay, we're going to move to cell function now, cell function. And we kind of started cell function on page 48 and 9. And actually, this is what we just did last Thursday. This was our most recent laboratory activity on cell function. Now, I'm going to defer question number 1 to lecture notes today because this is what we're going to talk about in lecture today. So to save time, I'm just going to defer you. That means put you off, and you have to look these things up in your lecture notes. We're talking about them today. Okay, so you might want to put C lecture notes, chapter 5, or something like that. Okay, so we're not going to do number 1 in lab today. But I do like number 2. This is just logical. And first I want to tell you, before we get started, what this word right here is. Auger. Has anybody ever heard of it before? Auger. Okay, so it's biology jello. That's what auger is. You guys know jello. Well, that's a trade name. We're really supposed to say gelatin, but everybody calls it jello. Okay? And biology jello is this stuff that we start with. It's powder. It comes from seaweed. It's powder. And we add it to hot water, and the water dissolves it. And then we pour it into different containers, and it solidifies. So that's why I mean it's like jello. Because everybody think about jello. Don't you start with a powder? Have you ever made jello? I know you've eaten it, but have you ever made it? Alex hasn't. Okay, so Alex, you can ask anybody who's made it. You start with a package of powder, and you dissolve the powder in hot water. Then you add some cold water to start cooling it. Then you pour it in whatever you want it to be in, and guess what? It becomes a solid, a fun solid. Okay, it's jiggly. Okay, we all love it. Okay, so this is biology jello. Why would you have this? To grow microbes on. And then we're also going to use it, you and I are going to use it later in the semester, because we've figured out that we can separate molecules using it also. So you and I will actually be using this later this semester. So my point is, agar is like Jell-O. Okay, so if you don't like the word agar, just think Jell-O. Okay? All right. In which would diffusion be faster? Remember, diffusion is the movement of particles from high to low concentration. Dye in water. How come? Excellent, Alaska, because water is less dense. It is less dense. That's excellent. So it's easier for the dye molecules to move through the water because the water is less dense. Everybody okay with that one? That's a great question. Okay, number 3 has to do with the activity that you and I did on page 48 and 49. And this is where we made this little bag last week with dialysis tubing and a kite string. Do you guys remember it? Now, the one we did, we put starch in the bag and iodine in the beaker. But I changed it up here. Everybody take a look. This is not on your review sheet. I changed it up here. Everybody take a look at what I drew. Anybody recognize my beaker? I know I'm really good at art. Okay, so I put water in the beaker. And then I put, does everybody recognize my drawing of the little baggie? Shake your head. Okay. I put molasses in the baggie. Now, do you know what molasses is? What is it? It's like real thick syrup. Real thick. So there's a bunch of sugar in there. Lots of sugar inside the bag. Okay? What's going to happen if you just do that and let it sit there for an hour? What's going to happen to the bag? Not sure? Here's what I think is going to happen. I think this is very concentrated in here with sugar, and there's no sugar out here. Look, concentrated here, but not out here. Say it again. It's not drawing. Let me try again. I don't know why it's not drawing. So it's concentrated here, not concentrated here. I can't write all that out. So I think water's going to move. What I think is going to happen. Now, would water move out of the bag or water move into the bag? It'll move into the bag. How come? There's way more water out here than inside the bag. So what's going to happen to the bag? It's going to swell. Water's going to move in, and the water will make the bag swell. So we could use a word to talk about the environment the bag is in. I remember this from last week now. Yeah. Hypo. Excellent. This is a hypotonic environment. This one's hypo because hypo makes cells swell. But this isn't a real cell. It's a pretend cell, but it would work the same way.
So are we supposed to focus on which way the water molecules are moving, or which way the substance is moving?
Well, we could talk about the molasses also. So what Emily wants to talk about is, what about the molasses, Ms. Steinke? If you let it set long enough, so much water will get in there, it will dissolve the sugar into small molecules, and then sugar will start leaking out. But that'll take a couple days. So initially what will happen is the bag will swell up. And then over time it'll leak back out, you're right. I like your thinking on that, Emily. Okay, take a look at my drawing. What's going to happen now? Like after an hour, what's going to happen? The water's going to leave. So the bag will get smaller, right? Shrivel, shrink. Like that? So what kind of environment is this? Hyper. Good. So that's the idea again on a hypertonic environment. Look how concentrated the environment is. It's got lots of stuff dissolved in it. Hypertonic environment means lots of stuff's dissolved in it. Hyper means lots, high. Are you okay on my little drawing? So if you're not sure, just read the description and on the back of the Scantron, because it's only 50 questions, so you're not going to use the back of the Scantron for any multiple choice questions. Everybody with me so far? Draw it out. Read the words and draw it out, and then think about it. And think about it. Okay, because these are thinking questions. You just got to think about it. Okay. All right, so let me talk about this question, number 4. I like this question too. It's a good one. First of all, on your piece of paper, amoeba is in italics and it's capitalized. Did you see that? What's that mean? It's its genus. But see, I was handwriting this, so it's not in italics. So what did I do? I underlined it. Okay, so everybody's okay, amoeba is a genus name? So I just rewrote the question. Now, I drew an amoeba here. I did a good job too. I did. Now, what I have, in the arrow is called a contractile vacuole. And a contractile vacuole is a water-filled vacuole. Now, let me explain what happens. Well, first of all, does anybody have an idea what this would be right here? It's nucleus, which means this cell is eukaryotic. And then let me tell you that this is not a cell wall. Amoeba doesn't have a cell wall. So what would this outside boundary be? Plasma membrane. I'm just going to put PM here and N here because I'm lazy. Okay, and then what did I tell you that this circle right here with this arrow is? Contractile vacuole. Okay, so let me explain what happens with the contractile vacuole. Everybody listen. A contractile vacuole is an area where the cell sends its extra water, and then this contractile vacuole moves to the edge and spits the water out. And then the cell again loads up all its extra water in the contractile vacuole, and then the contractile vacuole moves to the edge of the cell and spits the water out. Over and over and over again. Over and over and over again. Okay, everybody with me? All right, so let's read the question. Considering the function of the contractile vacuole in the protist amoeba, what environment would you predict that it lives in? And I gave you the 3 tonicities. Hypo, for sure. And what was the clue? Because I agree with you, but what's the clue? It's constantly swelling. Now, what would happen to the amoeba if the contractile vacuole stopped working? How come? Has no cell wall. It's very good. But okay, on question number 4. It's a good one. Just a side note, amoeba does live in a freshwater environment, which is hypotonic. And what I mean by freshwater, you guys know freshwater, lakes, streams, rivers, ditches, that's freshwater. That's where amoeba lives. So it needs contractile vacuoles. Okay. All right, number 5. For real quickly, a page number for number 5 is page 53. In case you want to go back and look in your book, where did we do this? This page 53 in the middle of the page about potato strips. I did an excellent drawing of the potato strips up here. I know you can tell my artwork is excellent. Yes. And so we had one test tube that had very concentrated salt water and one test tube that didn't have any salt in the water. Everybody knows 0% salt water is distilled water? Okay. And then I wanted to know which one would have a stiff potato and which one would have a limp potato. So let's talk about this 10% saltwater solution. And everybody, that's a potato in there. Okay, if you took that potato out and felt it, what would it feel like? This one right here. It would be limp. And I'm going to go ahead and use the word we used last week. Flaccid, yes. The word we used last week, flaccid. What kind of environment created these flaccid plant cells? Thank you very much. This is hyper. This one is hyper. Okay, how about this next one, which is the 0% salt water? If you felt that potato, how would it feel? Stiff. And the word we used was turgid. Stiff or turgid, which meant the cells were really swollen. They had very high turgor pressure. Really swollen. Push hard. Hypo. Thank you very much. Hypo. You guys are really catching on to these words, hyper and hypo. That's excellent. Hypo. But okay on the potato strips. And one more time, it's page 53, if you want a reference page for that, page 53. Okay. All right. We have a couple definitions here I'm going to go ahead and tell you about. Page 51 is where I got these words from. Page 51. Crenation is where red blood cells shrivel. On page 51, figure 4.8, which picture is a crenated red blood cell? B. What kind of environment would cause crenation? It's a pretty salty one. What's the word we're going to use for that? Hyper. Good. Hypertonic environment would cause crenation. Hypertonic environment would cause crenation. Okay, this is how this word's pronounced. You could say hemolysis, but it's pronounced hemolysis, hemolysis, or hemolysis. And again, I found this word on page 51 at the top of the page. And in hemolysis, red blood cells swell, but that's not all. They burst. Very good. This is bad news. If this is happening in a person, this is very bad news. What's going to happen to this person? They're going to die. Yeah. What kind of environment could cause hemolysis? That's good, Karen. Hypo. Hypo. Okay, this last part of number 6 says, be able to recognize blood placed in each solution. So we did this on page 51, table 4.4. I want to remind you that the tube that was hypotonic, which I think was number 3. Yeah, wasn't it number 3 was hypotonic? Okay, thank you for looking at your notes. That one looked different than the other two. I think you could see print through it. Do you remember why you could see through that tube? All the cells were burst. Yeah, there weren't any cells left. And we even proved it, I think, with Katie's group. Was it Katie and Diane's group? We made wet mounts and looked at those under the microscope. Yes. We even proved that there weren't any cells there anymore. And that's why you and I could see through it. But it's okay on that. Super. All right, turgor pressure. I'm going to go ahead and define it up here for you. It's the pressure that the cytoplasm exerts on the cell wall. You know, turgor pressure would not be a term you'd use when talking about animals. How come? We don't have cell walls. So when we talk turgor pressure, we're usually talking about plants. Anything with cell walls, though, you could talk about turgor pressure, because like bacteria have cell walls, or fungi have cell walls, but not animals. We wouldn't talk about that. But okay, on the definition of turgor pressure. All right, great. Okay, I have a sentence up here. You help me fill in the blanks. A turgid cell would be caused by a blank solution. Hypo. Because what does turgid mean? Limp? No, what's turgid mean? Real good and firm. Yeah. I have a turgid cell would be caused by a hypotonic solution. How about this sentence? A flaccid cell would be caused by a blank solution. Good. Hyper. It would make it shrivel and become limp.
What is a flaccid cell?
A flaccid cell would be a limp cell. Easy question number 8. Why would an animal cell but not a plant cell burst when placed in a hypotonic solution? Because of why? No cell walls. Animals have no cell walls. But okay on that. It's excellent. Okay, lastly, guys, real quick. I'm going to do this real fast because you guys know this. You know an acid, you can look up this in your notes, but an acid is something that gives off hydrogen ions. It's just out of your lecture notes. You know this from studying last week. An acid gives off hydrogen ions. Like we use this as an acid, HCl. Yes? Okay. A base accepts hydrogen ions. Again, this is just a review. Next week, this is what we're going to be using for a base, sodium hydroxide. In our laboratory work next week, we'll be using this base, sodium hydroxide. But lots of things are bases. Ammonia is a base, potassium hydroxide, bleach, that's a base. All right, pH scale goes from 0 to what? 14. And as you know, 7 is in the middle. What do we call 7? Neutral. How about if you're below 7? Acid. How about if you're above 7? Base. It's okay on the pH scale? Now, if you'd like a page reference for these things, it's all in your lab manual. Page 54 is... well, that's a nice picture, too. Figure 410, the pH scale is done very well there. Very, very nice. If you'd like a picture on that. All right, what's a buffer? Okay, so it's a chemical that it neutralizes, the best that it can, neutralizes pH changes. That's good, Jordan. It's a chemical that neutralizes pH changes. That's what a buffer is. And we kind of played around with some buffers on page 55. Like at the top of the page, we literally had buffer in a test tube and added acid to it. And I remember you guys said the pH didn't change much. And then we tested cytoplasm, and it didn't change much either, which shows that cytoplasm is buffered. Okay. And then I think that you guys may have actually drawn this at the bottom of page 55. Did you? Did you guys draw this? And I was telling you to match up which one of these would be a buffer and which one wouldn't. So everybody take a look at my two graphs right here. We can just call this graph A for fun. We can call this graph B just for fun. Which graph would you think would be a buffer? A? B? Make up your mind. It's B. Yeah, how come? Because it's resisting. That's very good. You could think of it that way. The chemical is resisting pH change. As I add acid, it's resisting. Finally, it's overwhelmed and the pH comes down. This one on letter A, this chemical didn't resist at all. It just gave up. Okay? No resistance over here. So letter B is your buffer. Letter B is your buffer. So this kind of goes with question number 3. Would you expect a curved or straight line graph of pH values using a buffer? You'd expect the curved graph, the curved one. Why? Because the buffer's resisting the change, as Katie Messina said. All right, our last question is, what's the function of an antacid? Like Tums or Rolaids. What's the function of it? Like, why do people eat those things? Why?
Reduce the acidity.
So it buffers the acid of the stomach. I remember Gosh, which one did such a great job? Alka-Seltzer was really performing well for us last week until we busted the test tube, but that's not the point right now. And then who else did really well? Did we have like Tom's that was doing a good job? Yeah? Okay. All right, so I want to just tell you real quickly that